We're very pleased to have a guest customer presentation today from Sean Anderson from the Vancouver Prostate Center in BC, Canada. He's from the Laboratory for Advanced Genome Analysis there that uses both array-based as well as next-generation sequencing platforms to support their clinical research to identify drivers of cancer progression and actionable targets in the development of novel therapies. Sean actually was part of one of the early access beta users of Nexus copy number 9, and he has done some great work using the new BAM multiscale algorithm in Nexus copy number 9, and he will be talking about his projects here. We are ready to begin, so I'm going to hand it off to Sean, and we can start. Sean?
Okay, thank you very much for the introduction, and also thanks to BioDiscovery for letting me get an advanced version of Nexus 9 to evaluate. It was really useful, and I wanted to show a couple of how the multi-scale reference tool especially was incredibly helpful. So today I'm just going to talk about some of the challenges and solutions to copy number estimation from whole exome sequencing in tumors. First, just a little bit about us. I had a pretty good introduction there already, but the Vancouver Prostate Center is located at Vancouver General Hospital. It's affiliated with University of British Columbia. We have about 21 research and clinical scientists. Our director here is Dr. Colin Collins, who is actually one of the pioneers of paired-end sequencing. So we've kind of had the benefit of growing up with NGS as it started, which has been really helpful. And it's been nice to have Nexus with us along for that ride. So again, there's a little snapshot of some of the platforms we have in-house. I think this is actually a little bit outdated here, but it gives you an idea of kind of the range of things that we do at Vancouver Prostate Center. So moving on, I just wanted to specifically talk about the advantages of the new Nexus copy number 9 version that's coming out. I was lucky enough to be able to get a trial version or a beta test version early on, and I was actually pretty excited about this because there's a couple of things that I really liked. One of which is the BAM multiscale reference for creating a pooled reference. And so I'm going to talk about these two things specifically in the context of the creation of a robust pooled reference in a relatively extreme case, and the adjustment of copy number baseline ploidy, and hopefully give some examples of how that's actually made a significant difference in the way that we're looking at some of these tumors. So I know Z-Way's already given a webinar on the multiscale reference tool that's now coming with Nexus number 9. So I'm not going to go into the nuts and bolts on this because he's already done it, but I just wanted to just touch on a couple things that I really think should be mentioned again that kind of make this stand out. So it's based on a read depth method that uses a pooled reference to generate pseudolog ratios based on reads. Pretty straightforward. But what's pretty neat is that it actually generates B-allele frequency plots based on reads at SNP locations, and I'm going to go over how important that is later with several examples. This pooled reference technique can be used for the shallow and targeted sequencing data. I haven't done shallow yet, but I've definitely done a lot of targeted, and it works great. And the whole genome, and it also works for obviously whole genome seq and whole exome seq with just a normal depth of coverage, which is going to be what I'm going to talk about today. And the last one here, I put a little star beside it because I think this is one of the coolest parts of this pooled reference bit is that it has an adjustable dynamic binning system. So this basically adjusts as it goes along the genome based on the depth of coverage. So the actual binning system changes depending on how deep or shallow it goes along the genome. So before I get into this too much, I'm going to go over the cohort that I'm going to talk about today. So the rest of my talk will be just specifically about these 21 samples. They're prostate cancer with what we believe to have no matched benign samples to go with it. So they're tumor samples, and as far as I know, when I got this, as I was told, it didn't appear that we had any normals to go with it. So again, 21 BAM files, 4 of which are primary tumor samples. You can see up here with the green arrow. 2 of the 4 are actually from the same patient. They're just from 2 like geographical, if you will, sections of the prostate. One's left transverse, right posterior. And there's 17 patient-derived xenografts, which are human prostate cancer tumor samples grown in mice. These were sequenced at Novagene on Illumina high-seq whole exome with approximately 200x average coverage. And if you see in the xenografts here an LTL in front, that's the name of our xenograft program here called the Living Tumor Laboratory that's headed up by Dr. YZ Wang. So one of the questions that people will ask sometimes is, well, why don't you have matched benign? Or sometimes people will say, why do you need matched benign? So it makes it incredibly. It makes it a lot easier if you do have a matched benign for each tumor sample, but sometimes you just can't get that. And one example is in a xenograft model. You really can't grow normal tissue in a mouse. You need that sort of driving tumor force for it to work. That's what the model was based on. So if we have the accompanying blood from the patient, that's great, but we just don't always have that. Sometimes these are a little older. We have degraded blood, or if the actual sample has changed over time, the blood wouldn't really match anyway. Another reason is a matched benign sample may be unusable due to poor quality. This occasionally happens. It tends to happen more obviously with FFPE over fresh frozen. There's several reasons, but even something goes wrong during sequencing, and you don't have a matched benign for that. Cost. So in the case of you're doing a large cohort, if you actually have 2 for every tumor, so for every tumor you need a matched benign, you're essentially doubling your cost of the run. So this can get obviously very pricey, especially with whole genome seq. And in some cases, adjacent benign is just really difficult to acquire. One example is in bladder cancer, where I guess there really is no adjacent benign. It seems that the tumor profile is ubiquitous or pervasive throughout the entire bladder tissue. Excuse me. So why is this particular cohort I have for prostate cancer of patient-derived xenograft samples especially challenging to make a pooled reference for? So I specifically for this talk and to show or sort of showcase this new multiscale reference tool, the new Nexus 9, I wanted to pick basically the worst example I could, or the toughest example I could, and show how something like this is robust enough to actually work, where in previously I might have been up the creek on this one. So this is sort of one of the things I was most excited about. So why is that prostate cancer PDX sample so difficult? A human tumor grown in a mouse removes most clonal heterogeneity. So these samples are incredibly homogeneous. And approximately 50%, possibly, in some cases more, of prostate cancers have an early stage recurrent copy number signature at chromosome 8, which is a loss. You see on the right-hand side here, I've got a little graphic of this, is a loss at 8p and a gain on Q. So you can see sort of between these 2 situations here, it can be very difficult to create a pooled reference. And the reason is, and I would even call this a bit of a caveat to using a pooled reference, is if the reference file stores the median value from the pool of BAM files, that's fine, so long as not more than half of the BAM files have the same copy number event, gain or loss, in the same bin. So you can think if everyone in our cohort has this exact same situation, an 8, and there is that possibility that a majority will, and it's homogeneous, so we don't really have things moving up and down in either direction to create an approximated normal, then this could be extremely challenging. So when I first looked at this cohort, I started to pull it apart. I really did cringe. I thought, oh God, this is going to be tough. So what I did is I decided to go with sort of a step-by-step iterative approach here and think, okay, I'm just going to try each one of these things and start with the easiest and just work my way through. So the first thing that I always try, if I can, just because it's basically the low-hanging fruit, is to try using a previous pooled reference and as similar as possible. So if I have recently done a project or I have a cohort that was done at basically the same depth of coverage, it's whole exome seq as well, it's done on the same platform, so you've got all these things that are pretty much identical. There is a possibility that you can go and poach the reference from another project and stick it in there. It doesn't take long to try. You can just put that in depending on how many samples you've got. You can let this run for a couple of hours or whatever, and you'll know if that works. And that's probably one of the easiest things to do. In this case, because the pooled reference that I tried, it didn't work, and I'll show you that, I had to move on to number 2. So number 2 is create a pooled reference based on random selection of samples. And Nexus recommends getting ideally as close to 10 samples in your pool as possible. So the third thing is just how well did that work? And one of the ways that you can tell how something works is if you are pretty familiar with the cancer type that you're looking at, and you should have some commonalities in there or just some signatures that you're used to, that if there's something fishy going on, you should be able to pick it out. Number 4 is once you've done this random sample selection that you put in your pool, I'll refine those candidates, because once I can start to look at these guys, if I see anything that maybe should be pulled out of the pool or something that's not really behaving, or we just have everything, the reference doesn't work because everything is so homogeneous, then we really want to start to put things in and out here to see if we get the best possible result, and I'll explain that. And again, in this case, I'm basically hunting for the closest to benign as possible, like a flat sample. Barring that, I'm going to use the most heterogeneous sample in terms of copy number aberrations. That if there's a gain in say 5, then I want to have a deletion in another 5. So at least I'm coming somewhere close to diploid in the pool. And 5 is to reprocess these samples each time with the new pool, and then check, and you'll start to get an idea just looking at these profiles if they worked or not, and I'll show you what that looks like. So this was the example of trying to steal a reference that I'd made that was incredibly successful on a bladder cohort. So I was kind of curious, can I, I've done this before with other things, can I take a bladder cancer cohort and just pooled reference and switch it over for this? I knew it was going to be a bit of a long shot, but I thought, hey, why not try it? Doesn't take long. So I took one of my primary samples, which is 1143LT, and I ran this against a very successful bladder reference that I'd used in a project recently. And it was almost exactly the same in terms of the platform was the same. It was Illumina HiSeq, it was whole exome seq, and the coverage was approximately the same. And then as you can see here, it really did not work. It's pretty messy. There's a lot of deviation. We really can't see a clear copy number signal in here, and the B allele frequency looks like it's basically barren. Or just looks flat. So let's move on to number 2. So here I'm just grabbing. I don't have any knowledge of this yet, of this cohort. So I don't know what these guys look like. So the best way to do it is let's just grab 9, as close to 10 as possible. I grabbed 9 of the 21, and I just made a pool of them. I just randomly selected them, and I threw these in the pool, and I tried it, and I ran them against another primary here. And when I did, it actually doesn't look too bad. It's usable. I'd like it to be a little bit better, and I'm not sure if this is, if there's a bit of noise in here, but as far as getting a first pass of this goes, this isn't too bad. And if I look down at the B allele frequency plot in the bottom here, I'm definitely getting a cancer signature with some allelic imbalances and some losses of heterozygosity. So it's kind of what you would expect, but I know I can make this better. I've got a hunch that we can do this, so we're going to try and refine this. So this was taking that random pool again with another primary. This was actually the second part of the same patient on the second location on the prostate. So we know the one side was definitely cancer, but when we look at this guy, we notice that there's a couple of strange things going on here. And I'm going to go back to talk about that chromosome 8 that we know about prostate cancer, where at least 50% of early prostate cancer, or early on, they adopt this recurrent aberration pattern where you have a deletion on P and a gain on Q. But in this case, we actually have the inverse situation. And this is what we would see if we ran a normal tissue against cancer. So we kind of flipped it around. So if you ever took a prostate cancer tumor sample and ran it against its matched benign, but you did it backwards, this is what you're going to see. So this chromosome 8 point here. And not only that, so that's sort of a tip-off that something funny is happening. And then looking down below in the B allele frequency plot, you can see that this is heterozygous, and it right in the center. And this is basically what you would see in a completely benign flat sample. So I'm kind of like, okay, this is a little bit strange, but it is a good sign, because it means that there's a possibility if we found one guy that's potentially flat in here, this could be about as close to a benign as we could get. And we're going to want guys like this in our pool. So what I do is, on my knowledge of or just knowing about this aberration on chromosome 8, and when it's inversed, the chances are that it's a flat sample that we're looking at, I want to select for that. So the best way to do that with Nexus is to go into the population view, and I hit the sort button up on the left here, and then I go over and I find in my frequency plot at the top, and ideally, in at least 50% of the cases, I shouldn't be seeing a gain on chromosome 8. Here's a blow-up version of it here. I shouldn't be seeing a gain on the P arm. So I want to try and select for those guys and look at them, because they could be potential flat guys. So when I click on here after hitting sort, that's going to sort that particular aberration, inverse aberration pattern up at the top here. So once I kind of have these guys isolated, I'm like, okay, I want to look further into these guys and find out just how flat and usable they might be. So I'm going to look more in detail on these guys, and here's a few of them. So 3 at the top here, you can tell, kind of look like what we were looking at before. If you can see it, it's essentially flat. It shows the inverse situation that we should be seeing in prostate cancer, which is what you would expect if you were running a flat, a benign sample against something that did have tumor content. And then the second big tip-off here is just looking at the B allele frequency. It's basically dead flat right across. We don't see a lot of aberration. We don't see a lot of loss of heterozygosity, and we don't see a lot of allelic imbalances. It's basically 3 straight lines. So this is kind of a, we can basically tell this one, the second one, the third one, all basically the same situation, with with the exception there's a couple little things here and there. I'm not too worried about those. Those will probably get filtered out with the median. But if I look at the bottom here, 352, this guy did have somewhat of that inverse signature on chromosome 8. He had a gain there. But when I look at his B allele frequency, it's not like the others where it is dead flat. It looks like there's more going on with this guy. So I'm a little skeptical of having this fella in the pool. So let me blow that up a little bit more. And now you can, and this is my I would say, check these things very carefully when you're putting things in and out of the pool, or you're putting things into the pool reference. You want to make sure that they're as close to what you're looking for as possible. And in this case, I've decided to throw this guy out here because he does have a pattern. It's definitely a cancer pattern. And even though he doesn't follow like that 50% rule that says that there's an 8p loss and an 8q gain, and you can see on chromosome 8 he has that gain up there. That's in fact, that's real, and that's just how this person's tumor profile looks. And you can see that's corroborated down in the bottom here. There's definitely some allelic imbalances and loss of heterozygosity. So I decided we're going to take this guy out. So right now we've only got 3 guys that we're going to put in the pool here, and I thought there's no way this is going to work. You want to get as close to 10, but Once I did it, I ran that same sample, 352, against those other three, and it's cleaned up significantly here. I'll come back to the B allele frequency of this guy later on, but I just wanted to show that it cleaned up, but not only did it clean up, we have CGH data from this patient from a while back. Granted, there has been some changes over time in the mouse model, but looking at the old CGH data that we did from the Xenograft that arrived from this patient in the past, you can see it's basically recapitulated in the whole exome-seq that we've done. So we know that we're kind of hitting all the right points here in terms of capturing the copy number profile. And I'm not going to go into super detail on these, but I basically do like a piece-by-piece aberration here. And indeed, the whole exome-seq actually works pretty good here in basically reproducing what we've seen before. And I guess like chromosome 4 is a good example here, where we have two little homozygous deletions and a single copy loss here in that pattern. You can see pretty clearly right here. And there's another homozygous deletion arrow dip right in the middle focal. So moving on and going back to actually 1143, which is one of those primaries, when I run him now against the refined group, the refined pool, where I take those three that we're really happy with because we think they're flat, and I run them against this guy, I can see that in fact this is a benign sample. So it's dead flat in copy number across the top, and not only that, that's corroborated by the B allele, excuse me, the frequency plot. So you can see that this is dead flat as well and heterozygous in nature. So well, I actually went back to pathology and asked them, hey, I've got these two primary samples, one from one side, one from the other. And in fact, under a closer examination, it looks like this sample was in fact adjacent benign tumor looking through the microscope again. And I wasn't quite sure actually if they had done that on purpose just to see if I could find it, but I'm assuming it was just a mistake in sampling. So now we want to go back and check and see how this refined pool worked with some of the other guys in here and some of the more complicated patterns that we saw before. This is another Xenograft 313HR. So this is what it looks like with the 9 random samples in the pool. Now this is what it looks like with the refined. So you can see I'm just going to go back and forth here. So you can see how much that gets cleaned up, which is obviously incredibly important. With the tighter profile, you've got a greater chance of well, seeing the actual biological situation, but also being able to pick out more focal aberrations because you're getting a less deviation from probe to probe. So another example is 352 that we looked at before. This is with a pool of 9 randoms, and now with my refined guys, much tighter. So quite happy with this. So there, I played around with the pool a bit, and I jumped ahead to show you like basically once I'd refined those 3 guys, and yeah, so I'm actually quite happy with this. And there's another example here of another Xenograft 539. This is again with the 9 random samples, and now I'm going to compare it to the refined. And as someone that does copy number, you'll probably appreciate that when you get something like this, you're pretty happy. This looks great, and you couldn't ask for much more. So we know that worked. So now the next thing I want to talk about that's a definite benefit of this Nexus copy number 9 version with the new multiscale reference is the adjustment of copy number baseline ploidy. And this has been sort of the thorn in the side of looking at copy number for a long time, especially in the CGH days. So barring doing other experimentation with, say, fish or STISH, it was very difficult to see certain situations where you've got this diploid line that has moved in some sort of direction, especially in a balanced situation. So I'll get a little more into that. So what I wanted to say was that when you're processing these samples with the BAM multiscale reference tool, it creates the copy number plot with the corresponding B allele frequency plot as we've seen underneath. And what that is, it allows the user to recenter or confirm the copy number profile at the top. So you've got them right there side by side. So you basically got this like correction method that's automatically right there, and you can verify what you're seeing or make corrections to it. And this is especially useful in a tetraploid balanced occurrence. And I'll give an example of that and how much this is basically advanced copy number accuracy greatly compared to say 5 years ago or 6 years ago. So looking at that sample 352 is a bit of a guinea pig for us here because it was especially difficult. If we look at the copy number plot at the top and we compare it to the B allele frequency plot at the bottom, we start to see there's, things just aren't really adding up here. And so before I get too deep into this, whenever I want to start doing these comparisons, the two things that I kind of have in the back of my head is allelic imbalances, which you usually see around these two lines right here, should correspond with gains or amplifications in the same spot up above in the copy number plot. And the converse of that is loss of heterozygosity, so fellows around this area here, should correspond with singular homozygous deletions up in the copy number plot. And here I'm going to point some out. So you can see, based on what we see down here, first of all, let's look at chromosome 4. It looks like there's a single copy loss here. But if I look down in the allelic frequency plot, it looks like it's saying basically that this is heterozygous, and we've got this great little region of heterozygosity. Well, that just doesn't make sense. How can you have a single copy loss and have both heterozygosity situation here? And then I've got 4 samples with green arrows here, or 4 examples of allelic imbalance, one here, 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 and those correspond to, if we look up and we follow this, they're basically diploid, or they're being called diploid in the copy number plot. And I mean, this is, I think the algorithm that Nexus uses here, it's almost impossible to find these diploid lines. I mean, it does the best that it can, but it really doesn't know where to center these things, especially if something biologically strange is happening. So what I've done is moved that up. I'll just go back and forth here. So you can see right here, if you keep your eye on chromosome 4, I've taken that single copy loss, and I've bumped him up. So now he's up on the diploid line here, and now you can see that this particular sample has been corrected via the B allele frequency plot. And I'll show some more examples of this, but back in the CGH days, this would have been very difficult, if not impossible, to do without some sort of other verification or validation study. So going back to another particularly difficult sample for us in the past, we actually signed off on this one back in 2014. So this is a Xenograft in CGH at the top, and done recently with whole exome seq PDX on the bottom. So we can see that the profiles basically recapitulated, which is great. So we know we got the right patient. Everything looks fine. The calls are relatively the same. But one thing that we noticed is back in 2014, when we finished this, we thought, hey, this is great. We're done here. This is good to go. We've got 5 copy number states. We've got the homozygous loss, single copy loss, diploid gain, and amplification up in the top. It's probably centered around diploid. Bang, done. We're finished here. But when we run it now, as I just did like about a month ago, I ran that similar sample. This is a recurrent sample of this patient, and we ran it again. Now we have the B allele frequency plot with this new function in Nexus, and I can see, well, if you look at chromosome 6 here, we can see that, well, that doesn't really make sense that he would be heterozygous if we have it called as a single copy loss. It really doesn't seem to make sense. So that was the first sign there that we should probably be moving this thing up. So this is what it looks like if it's been recentered. And here's chromosome 6 now, which we have I'll switch back and forth here so you can see. Before it was down as a single copy loss here, chromosome 6. I'm just going to go back. And now that's been recentered to the diploid line. So what's actually happened in this particular case is that we've seen the entire genome has been duplicated, and this is actually a tetraploid situation. And I use Nexus just by actually grabbing that call line here, putting in that location in diploid here, and then just hitting apply, and this moves this up for me in one foul swoop. Excuse me, which is extremely valuable. Now, especially when we go back and we look at some FISH images, and I don't want to go too far into this, but one thing I wanted to just quickly mention is on chromosome 10, there's a gene called P10. Before we corrected this, we had homozygous deletion that was quite low down here, so it would have been somewhere down around here before, and now we've moved it up. P10 is somewhere up around a single copy loss, which makes a lot more sense now when we go back and look at some FISH data that we've done back around 2015 that showed that we did have some probes lighting up for P10, which would basically be impossible if it was a homozygous deletion situation. So now that we have this, we've confirmed that this is tetraploid by the B allele frequency, the data is really starting to make sense now. So that's pretty much it. I just want to summarize what I've said here. So Nexus copy number 9 with that new BAM multiscale reference has proven to help me overcome 2 recurring challenges that we've had in our analyses. And so the 2 things that I hope I showed today was that the creation of a robust pooled reference by a step-by-step method for a particularly difficult cohort. I was pretty scared that this wasn't going to work at the beginning, and just by sort of attacking this piece by piece, I think it worked out really well at the end. And I did that by looking for heterogeneous or non-recurrent aberrations or flattish samples in order to refine my reference pool. I really went on the hunt for flat guys on this one, and it really paid off. And that created an accurate copy number profile that, luckily enough, we have previous ACGH on some of these patients from PDX models, and we were able to confirm them, so we feel really comfortable about that now. And the second thing I hope I was able to show you was the adjustment of copy number baseline ploidy. So using that B allele frequency plot allows either the easy confirmation of what you have in your copy number plot above, or allows you to recenter to that diploid situation in the copy number plot, as we saw. And that lets you go back potentially and compare to some of your old data, or in our case, I'm now reevaluating some of the previous data that we did before, and you saw that tetraploid situation, which we just didn't know before. So I guess with that, I'd like to thank you for listening, and I'd like to go ahead with any questions. Thanks.
Thank you very much, Sean, for the really nice presentation. And we'll head over to the questions now. Would you expect your results to improve if you had more flat samples for the reference or included more normals?
Yes, it is that definitely the more the closer to 10 that you would get, excuse me, with flat samples, definitely it would probably clean things up a little bit. But I think by even just using those 3 flattish samples, I was actually pretty surprised. That's incredibly usable for all intents and purposes. And if I compare to the previous ACGH data, I didn't drive in and zoom into any chromosome views, but when I looked at them, I was actually incredibly surprised how accurate like focal changes were. So yes, always more is always better. But in this case, yeah, I think I was very happy with the way that those 3 flatter samples worked. Thanks.
Thanks. The data you showed had a pretty high coverage level. Have you used other data with lower coverage level successfully with the tool?
That's a good question. Yeah, we have around 100, around 100x. We've done a few of those. But as far as using the low pass or like a shallow sequencing thing, we've definitely done targeted with like Oncomine panels and some custom panels. But as far as the really low shallow sequencing, we haven't really done that yet. That's probably the next thing I'll be doing coming up this month with some renal clear cell with some kidney samples, and we're actually pretty excited to try that. But no, I haven't done too much with low pass yet.
Great, thank you. And I would just like to add to that we actually have tried it, the algorithm with less than 1x coverage, and it has worked. And we do have a white paper describing some of these results. And at a recent AACR meeting, one of the customers actually presented some low-pass sequencing data where they'd used 0.5x. This was with the previous algorithm, but it should be working just as well with this one as well. So the next question, can you use the multiscale reference tool on other captures, such as genome panel?
I'm not sure what you mean by genome panel.
Genome sequencing, targeted panels. I think you showed exome, whole exome sequence.
Oh, sorry, yeah, like whole genome sequencing, and yep. I would imagine you should be able to, and I'm going to go ahead and say maybe Z-Way or you might be able to answer this better, but I would, my gut instinct on that one is yes, it should work because of that adjustable dynamic binning that takes into account the difference of the depth of read along the genome. So in the case of whole genome seq, you've got so much to work with there that, I mean, the adjustable binning is obviously going to be beneficial, but in the case of targeted seq, that's what's really going to be beneficial because your off-target regions on sort of that backbone area where you're going to do your core spinning is going to be incredibly useful. And I'm just trying to think if we used a pool reference. I don't think we did for our last OncoMine targeted panel, but I don't see why not. I think that just adjustable binning should answer that question.
Great, thank you. And yes, we have used it on the other types of captures as well as we've had customers use it with targeted. Actually, the Multiscale Reference, another early access user, presented their data at the recent AACR meeting as well. So they were very pleased with the results on their targeted panel data. Another question, did you confirm the NGS results with array results for each sample?
No, some of we had new PDXs put in there, so we only had confirmation with about what was it, maybe 6 out of the 21 that we had. Oh, and actually that primary was done in a PDX as well. So yeah, I'm going to say like maybe 7 or 8 we had array CGH for. But again, those were done on a different passage, so they were the same patient, but put into it was a different mouse. So the patient signature is still there, but the whole exome seq, technically it was a different mouse and different tissue. But yeah, I'm going to say 7 out of the 21, I believe, that we had ACGH for.
Great, thank you. Here's a sort of a long question here. Your comparison of the data from the same sample, a recent data set and a data set collected 2 years ago, show new CNVs not seen earlier. Is this a cell culture, same patient, 2 specimens taken at different time points or artifacts?
Yeah, no, that's a good question. So I don't know if in the title you can see there, and when I looked at 313H, that sample, the one that I did back in 2014 was ACGH, was 313H, and the next one that I did, or the one that I did in whole exome seq just earlier this year and finished about a month or 2 ago, that's 313HR. So it's actually a recurrent version of that sample. So the basically that's a more advanced form of the cancer. So the tumor profile is essentially the same, but yeah, you're absolutely right. That's a good eye to pick that out because there are some new things, and that's sort of the bread and butter of this project, is to look for those new things. So that was a good observation to catch that. Thanks.
Great, thank you so much. That is all we're going to cover right now with the questions. I am going to close the webinar. Thank you again very much, Sean, for the lovely presentation. We will have a recording available for those people who may have missed part of it or would like to review it again later, and we'll send out an email when that's available online. Again, thank you everyone for attending the webinar.